Hello, everyone, and welcome to the latest episode of our ALS Clinical Trials Unboxed. My name is David Taylor. I'm the Vice President of Research and Strategic Partnerships at the ALS Society of Canada. We are joined today by two individuals who are very excited to hear about their clinical trial that is ongoing and expanding, hopefully in the future, around a particular probiotic called HA-114. It's a great story of going from lab to clinic, and here within Canada, Canadian innovation going straight through to amazing clinical research happening right here within our borders. So we are joined by professor at Université de Montréal, CERCHUM, Dr. Alex Parker, and the head of the clinic at the Université de Montréal ALS Clinic, Dr. Geneviève Matte. We're really excited to get into the questions afterwards, but without further ado, I will pass it over to you to share your screen. Thanks for joining us, and we look forward to the talk.
Thank you for the introduction, Dave, and thank you guys for giving us a chance to talk about this project we're working on, because we'd like people to know about it. It's ongoing, so if you have any questions, now's the time. And it's about, as Dave said, a probiotic approach perhaps for ALS. And so I'm going to try to be a little short and snappy about like how we got here to understand why we're doing this, what we're looking for in a mechanism, how we kind of translate this to a clinical trial. And this is a generic or general ALS introduction slide, but we don't really need to talk about this today, other than the fact that, the hard work of many years of genetic studies has given us a list of genes that are linked to ALS. And for people like our lab, these are useful tools to try and build models. So if we can build a model, then maybe we can find a therapeutic. It's really like that. We simplify it a lot, and by simplifying it, we really simplify it, because us like other labs, we use animal models, but the animal we use is a worm. It's C. elegans. It's a very famous animal in terms of laboratory research for genetics, studies in aging and things like that. And it's a great model because it has a very simplified nervous system, and you can use it to hopefully find things. And basically, the approach we've used for several years now is that we take genes that are directly linked to ALS, we put it in the worms, and forgive me if you've seen this video a million times, and express it in the motor neurons. The worms are not happy, they don't move very well, and this is an important phenotype because it's something we can use. The whole goal of this is to try and find things that correct a movement problem, and secondly, the animals also show motor neuron degeneration. So what we're trying to use these very simple animals to screen things, to look for molecules, drugs, whatever, that might be protective and would warrant further investigation in more complex models and, more importantly, the clinic. And so what is the relevance of C. elegans models to ALS drug discovery? So I thought about this yesterday, and some of the talks coming out of the recent Congress here in Montreal, and I thought, well, work starting in worms was the basis for leading to the pimozide trials, to look at mechanisms of all synaptic activity, neuromuscular junction function, the peripheral nervous system as a target, perhaps targeting SARM1. This is an area of investigation in clinical trials, and we did some work in this early on in the days using worm models. And more recently, we collaborated with Justin Achita's group, looking at PIC5, targeting PIC5 as a mechanism for motor neuron generation. So we like to think that a simple animal, even like worms, might be useful to find new therapeutic approaches. And the thinking for this came from the fact that even for ALS, there is still great variability in like the age of onset, progression, and things like that. So perhaps there's something beyond just the genetic. What if there's an environmental component, right? And if this is true, what people are looking for all kinds of things: environmental stresses, exposure to toxins, etc. But one thing that's been gaining a lot of traction in the past couple of years now is, what about the contribution of the microbiome? Or a collection of bacteria and viruses, et cetera, in the gut that can influence many, many phenotypes. And what if the microbiome could in fact influence ALS phenotypes? And if this is true, in general, there's two ways to think about it. One is like, perhaps there's a group of, like, we'll say, for example, harmful bacteria. So you would like to remove those if those are affecting ALS in any form. But what if there are protective bacteria? things that are protecting the nervous system from degenerating during the course of ALS. Now, if this is true, if you could find them, then you could quickly develop them into therapeutics. And they would essentially be a probiotic. This is taking a bacteria which has potential protective effects, but here specifically for ALS. Now that's hard to do. How would you find these? There's lots of approaches, but using something simple like worms, as I mentioned before, we use it for screening, right? So you could just grab a collection of probiotic bacteria and just test them systematically to find which ones are like reducing these movement problems, causing the worm, extending the lifespan of worms perhaps, and more importantly, like stopping the motor neuron degeneration you see in ALS, at least in our worm models. So we were lucky enough to collaborate with our L'Allemand Health Solutions here in Montreal, and they provided us access to their collection of probiotics, and we screened them, right? We tested these bacteria one by one in some combinations, and luckily we screened them in our ALS TDP43 FUS models. They work in other models as well. And surprisingly, we found one that worked very well. And if you I'm just going to go through this quickly. It's like these bars that represent the red is neurodegeneration. And just by feeding the worms this bacteria, this probiotic, it decreases the amount of motor neurodegeneration in the ALS models by a simple change of diet. Because normally the worms eat one type of bacteria, we give them this instead, and it has a very strong protective effect. Now, this is interesting, but what about the mechanism? Because if you want to pursue this further, it would be good to have some information about what is going on, because this could be something you could track in a clinical study when you get to that stage. So this is a lot of work done by Audrey Labar, my laboratory, for many years now. And I'll just cut to the chase. What we think is happening is that there's a problem in mitochondrial energy production in the neurons, the motor neurons of our ALS models, perhaps patients. And not enough energy is being produced by the metabolism of fatty acids. And we think that the HA-114 is bypassing this problem and providing energy to the neurons so they can fight off degeneration. Right? And there's a lot of information here, but we can see that in our ALS models, in the worms at this stage, is that they have a problem in lipid metabolism. The only thing you need to be concerned with here is this little blue box where the worms are accumulating, the ALS, the TDP43 worms, they're accumulating lipids, which suggests there's a problem. We don't know what this, you can't extrapolate this exactly to humans, but there's a problem in lipid metabolism, and HA-114 can help bypass this. And so using the worm models, this is a summary of like a couple of years of work, is that there are fatty acids coming from the HA-114 bacteria that are protective. And if you start to take these apart and add them individually, it's less effective than the entire probiotics. We think the whole bacteria itself is more protective. The specifics have to do with mitochondrial beta oxidation, because if you start disabling these genes, the protective effect is lost. And we think that perhaps this pathway is impaired somehow in ALS models. And this could account for the overall motor neurodegeneration in patients and models. And so that's not it. That's not the whole point, though. Like, we want to validate this beyond worms. So we're at the same time in the laboratory, we're testing ALS mouse models. This is an ongoing project. And what we're doing here is treating, just giving these mice, SOD1 and TDP43 mice, HA-114 in their drinking water. And we're testing for all kinds of things at different ages. And we're in the middle of it. We got another nice big data set yesterday, but haven't had time to go through it yet. But what I can tell you generally is that from the SOD1 experiments so far, that the mice who've been drinking, ingesting the HA-114 have less motor neurodegeneration. And what we're also doing at the same time is taking a look at the lipid profiles. So we think that this might give us an idea of what's going wrong and let us get closer to a more specific mechanism. So what I'm showing you here is our lipidomics experiments with our collaborator, Mathieu Ruiz, where we're looking at, these are select lipid profiles of two models, SOD1, wild type SOD1, and SOD1 with the probiotic. And what you can generally see is that the profile of lipids in the disease model, the ALS model, is different than wild type, and giving the mice who have taken the HA-114, the lipid profile shifts back to a wild type normal phase. So this suggests that there's something going wrong with lipid metabolism in the ALS models, and HA-114 can push it back. Now, this is interesting because we see something similar in the TDP-43 model, but we can also see changes in the profile of lipids coming from the plasma, liver, and the spinal cord. We added cortex for TDP-43 for reasons. But this is interesting because on one aspect, this could be used as a biomarker, perhaps, but more importantly, changes in plasma lipid concentrations with and without HA-114 might be something useful to monitor in patients, and would also give perhaps an aspect of like target engagement with the treatment. So this was interesting because we thought perhaps if we could move this to a clinical setting, which we're doing with Geneviève, is to look at the lipid profiles of patients, because not a lot is known. It's an emerging area of research. And we can also check to see what happens in the patients who would take the probiotic, to see if it pushes it back to a more a wild type of thing. So that's the preamble for how we got here. We did this work where we identified a probiotic, and it's working in several animal models, and it gives us a chance to look at something relatively easily in patients, I suppose. But we've been fortunate that we're in collaboration with Laumond and our other collaborators, with support from the Weston Foundation. This is now part of a clinical trial here in Montreal. This is run by Geneviève, and I will now pass over you don't have a clicker even. I'll advance the screen for Geneviève for the next sections.
Yes,
So,
thank you, Alex. So I'm going to present to you the clinical trial, how we got there. So next slide, please. So thanks to Alex's work and success at grant writing, we were able to get the project funded through the Weston Family Foundation. And our goal is truly to bring the trial to several other sites all over Canada. We've been already recruited in Montreal since the spring of 2024. Next slide, please. So Alex came to me with these ideas about 6 years ago now, and with interesting findings. And the question was, how could we bring this to patients? And thank God, probiotics are fairly safe, we think, of an intervention, or easily manageable if there was something we discovered. So I thought that's a good way to start. And also, well, when someone comes at you with ideas from the lab, you have to think as a clinician researcher, does it make sense? And yes, indeed. So the microbiome and the the microbiome has been associated to neuroinflammation, and that's a theme that comes back once in a while in ALS. And there are reports in some series, the findings are not consistent, but we observed that probably there's a problem with the microbiome of some ALS patients. This still needs to be elucidated. Some researchers in Canada, such as Gerald Pfeffer, are interested in that. So the intervention of giving a probiotic to change the microbiome and have an impact on metabolism is very logical. We see also in ALS patients that there's some weight loss and catabolism, so that is the body degrading itself to get energy. And so the fact that probiotic improves this in ALS worms, then we can expect that perhaps we'll see the same thing in ALS patients. So, Alex, next slide. So that's how we designed a clinical trial. Ideally, we'd want, and you'd want to know if the drug works. Does it stop ALS? Does it slows it down? However, this takes a huge number of participants, several hundreds, and that's not exactly in our capacity for now. So, as Alex mentioned, we went for target engagement, see can we change the lipid profile. And then, as Alex mentioned, what do we know about lipidomic profiles in ALS patients? Well, not all that much. So that's why we thought it was a good primary objective for our study. For sure, our other aims are to see if there's a progression of disease. Is it different in patients exposed to the drug or not? We entered some cognitive and behavioral assessments. That is a part of because of the interest of our sponsor, but also because they're part of the ALS symptoms we should be considering. And we're looking also, does it change neurodegeneration? So we have some biological markers such as neurofilaments. So we're measuring this. And also we want to see, does it actually change the microbiome and have samples from patients from the start. And because we don't know much about the lipidomics in ALS patients, that's why we're assessing also healthy volunteers. And that way we can see if there's indeed a difference in the microbiome of patients at start, but more importantly, is the difference observed in worms between ALS worms and healthy worms is present? So is there indeed a lipidomic difference? And will the treated ALS patients get a more normal lipidomic profile after exposure to the probiotic? And as usual, well, we want to see if the intervention is feasible and safe and tolerable to patients. Next slide, please. So we're looking for 100 ALS patients diagnosed with ALS. We need caregivers because of the behavioral assessments. We need an observant, someone who observed the patient to answer. And we need also 50 healthy participants as the control comparator. The main inclusion criteria are having ALS and with a certain level of function, certain level of vital capacity. We're still a bit less restrictive than classical clinical trials. And also, well, there's the participants must not change drastically their eating habits or introduce probiotic in their food, their diet. So we don't confound the effects of the product we're trying. Next slide, Alex. So the exclusion criteria are use of respiratory support and other diseases. The indicated drugs for ALS, edaravone must be on a stable dose. And out of precaution, patients who are immunocompromised cannot be included. Next slide. So it's a placebo-controlled study. The ratio is 1:1. And we're using a placebo so that we have a comparator. We cannot only rely on feeling better or progression. We need to compare, unfortunately. We have to remember that in science, we test the null hypothesis. So that is, we start with the assumption there's no difference, whatever we'll do. But to disprove that, what is the size of the effect we'd like to see? And that's how we calculate the sample and what's needed to be done. We propose interventions that we feel will be of benefit to patients, but scientifically, our methods start with the assumption it's not going to change anything. So, but we have good pointers that it will here. So in terms of visits for participants, we try to keep it as close to clinic visits as possible so that sites may decide that the research visit is actually a clinic visit so that it's easier. So there's a screening just to make sure, screening visit to make sure you have all criteria to enter the clinical trial. And then there are 4 other visits at just about every 3 months over a 6 months period. So a baseline visit where we collect stool, the lipidomic profile, the neurofilaments, and then a visit 4 weeks later. That's more to check on safety and make sure the patient understands very well how to take the medication. And 2 other in-person visits at 3 and 6 months where we control again microbiome, the lipidomics, and neurofilaments. And this is going to be what the data will be comparing to the baseline to see as our treatment changed something. And there are interspersed follow-up phone call visits just to make sure the patient is well and there are no issues. The product itself is only a sachet, a powder that you mix in a liquid. Next slide, please. And these are our coordinates to reach out to us. The research probiotic is a address for patients. We're opening a site in Fredericton at the end of February, and we're looking forward for other sites elsewhere in Canada. Next slide, please.
Yeah, so this is the thank you slide for everybody who's been involved with this up to this point, because we can't do this by ourselves, obviously. And it's been an iterative process with great support, early from NSERC and then ALS Canada at different stages of this. And right now at the Weston, so it's been my tax. And so that's been good. And also the continued support from Loudmount Health Solutions has been invaluable. They've been really great about everything. So thank you everybody for all of that, and our collaborators doing the lipidomics, and here at the SHUM for the genomics, Martin Tetra. So great big project, lots of people to thank. But I think at this point we're ready to take any questions you may have. Thank you, Geneviève.
Thank you, Alex.
Thank you both very much, Dr. Parker and Dr. Matte. That was great. I think it's exciting. I can say, as having the good fortune of seeing the early days of when you proposed this, Dr. Parker, after just sort of serendipitously coming across this particular one and then seeing where this is now, that's really exciting. And it's a testament to both the science in ALS and also the ability in Canada for the clinicians and the basic researchers or the fundamental researchers to sort of have those relationships to allow these types of things to happen. So that's so cool. I have a couple. Please put anything in the question and answer box if anyone out there has a specific question. I do have a few practical questions before I get into any nerdy questions about the science, if we have time for those, because I think the practical questions are the ones people are more interested in. So first of all, for Dr. Matte, you'd mentioned about willing to be maintaining someone's diet and not introduce any probiotics. I think one question that might come up is, what if I'm currently someone who eats an Activia yogurt or something every morning? Do I just stop that? Can I still be involved? I don't even know if that's truly active culture bacteria. And then also when it talks about standardizing or keeping your diet the same, is there anything around the regimen in terms of like before the clinic visit or before a blood draw that they would have to follow specifically to sort of make sure nothing is changed too out of whack?
Yeah, so we ask patients to stop things like Activia. We'll take Danone's word for it that there is active culture in it. I buy another brand,
Okay.
and I can tell you it's inert. But no, we ask patients to refrain from consuming products that have a label probiotic, like, or not start and not go, things like kimchi, sauerkraut. Also things like not going on a keto diet while on
Yeah.
the clinical trial. Albrioza is now taken off the market, as far as I know, but um, this was a tricky part for us in that there's
Yeah.
the sodium phenylbutyrate might
Mm,
change the lipidomics. So we had found a way to handle it, and then when the product was withdrawn, we had to find another way to handle it. So we really try to have patients keep the same diet. Because of many unknowns, we do not well, having a peg tube is an exclusion criteria. However, during the study, if the patient needs one because it's clinically indicated, then it is allowed.
okay. Well, thank you very much. And certainly if people have specific questions around the things, they should reach out to the emails that were listed in the presentation. This will be streaming afterwards. So if you didn't catch those, you can definitely go back and find them on our YouTube channel. I know another practical question that comes up quite frequently is people will think, I would think the same thing is I could go to Pharmaprix or to Shoppers Drug Mart and I would look in the probiotic aisle and I'm going to look for HA-114 and I guess is there any way you could comment on what that would be versus like something that you might find over the counter or what's out there? And then certainly, the reaction that you see in terms of a lipidomic profile that's specific to a purified HA-114.
Yeah, so I can take this one. So this is a question we get a lot. It's like, can I get some? Can I go down the street? And it's like, no. So this particular strain is not commercially available by itself. HM14 is a product of Laumond, and it's been used in other other applications, but usually as a mix, right? So sometimes people will send me, I found this on some website somewhere. I'm like, yeah, but first, it'd be nicer if it was part of the clinical trial. Second is that we don't know how well it would work in combination with other bacteria. We have even like some indications from our worms that certain mixes dilute the neuroprotective effect. So that's something we don't know, but it's an open question. We're not sure how those things would work, and so we don't advise that. So that's the short and long of it.
Okay. So now I get to ask a nerdy question, which is when I first saw that this was a change in a lipidomic profile and sort of metabolism alterations that could be related to this. What I found kind of exciting was, we've learned in recent years that upstream of diagnosis in the sort of asymptomatic stages, there seems to be metabolic disruption in people with ALS. They've done this in studies of people who are like genetic carriers who who convert to the disease and et cetera. But it's really complicated, as metabolism always is, right? Very, very complicated as to what an actual signature would be. But after this trial, once you get some of that data, do you think that might also help you to be able to say, like, if you looked into some data on lipidomic profiles that existed from some of these asymptomatic cohorts, you might be able to see if there's any kind of changes that might reflect early treatment or early intervention with something like HA-114 that could actually have an preventative effect or a early asymptomatic effect on ALS?
Yeah, so we're hopeful this is what we will see. And I think one thing that's really been great about this current project is that we have all of these mouse experiments going on. We're treating them for early versus late. And even we don't have the full data set in now, but we start to see like a change, an early change, and it's progressive. And then we think that the HM14 is like converting it back. And what we're really hopeful for is like once we get to the end of this, we might be able to specifically map out each lipid, like each specific change in these lipid profiles back to very specific enzymatic metabolic processes and which step. We might be able to get close and say, look, this part here is what's going wrong early. And maybe that'll explain a lot. It's something we can look for. Or maybe even down the road, this could be another way to develop a more targeted type of approach. So we think that It's interesting because the lipids, is it confusing? I don't know. It's a complicated issue, but we think this for our very holistic, large scale approach with the mice. And then once we see this, and the data come from the patients and the other studies that are out there, we think we should be able to piece together what's going on early, during, and late. And I think this will just be valuable information no matter what. And it will certainly give rise to additional targeted approaches. We hope, one thing at a time.
I think it's amazing. And honestly, target engagement is the thing we're missing so much in ALS. So this trial is really hitting the mark in terms of what we need to be doing more on ALS trials. So really appreciate that and super appreciate your time. We're already out of time, even though I had so many more questions. But if you have questions out there or you are interested in this trial, please do reach out. We would love to see this trial get enrolled and flourish and be able to learn what we can about the value of HA-114 in people living with ALS. So thank you both, Dr. Parker, Dr. Matt, and look forward to seeing everyone again soon at the next ALS Clinical Trials Unboxed in March. And wishing everyone a great rest of the day. Take care.
Thank you.
Happy New
Thank you.
Year.